All right. Thank you very much for the opportunity to both attend and participate in DigiCrop this year. I'm going to be telling you a little bit about work in my lab that is focusing on better ways to prioritize which gene models in the genome are likely to have a phenotypic impact on the organism in question. In my case, crop plants such as corn and sorghum, maize and sorghum. So a lot of the work in my lab actually focuses on using new high-throughput phenotyping technologies and then using this to link genotype and phenotype. But this is actually a really hard thing. You require a lot of different disciplines to collaborate and play well with each other and understand what capacities they do and do not have. In longer talks, I go through a couple of those layers. Because this is a short 15-minute talk, I'm going to focus primarily on the quantitative genetics layer. So this is after sensor data has been collected, plants have been grown in a field or a greenhouse, sensor data has been collected from those plants, and that sensor data has been converted into numerical measurements of traits we are interested in about the plant, for anything from leaf angle to oil content of seeds to flowering day, what have you. Now, if you look at numerical trait data that comes from conventional phenotyping, a student going out into the field with a ruler and a notebook and making measurements, versus the types of data that come from high-throughput phenotyping data, there are a couple of real differences in pattern. One of those differences is that high-throughput phenotyping data is much more frequently time series than is conventional data. And I think the reason for that is basically economics. So each time I or my student go to the field and make measurements, it takes us about as much effort and about as much time. So we really have no incentive to go to the field and collect more than the number of time points we really need for our experiment. Usually, the answer to that is 1. With high-throughput phenotyping, a lot of the effort is just getting the technology to work. Let's take the example of a UAV. So we have to buy the UAV, we have to find someone to fly the UAV, they have to get trained and certified. Then we have to figure out how to process the image data collected from the UAV and extract numerical measurements from it. Once we put all of that effort in, the additional cost in terms of money and time and effort of just going and flying every week instead of once during the growing season, it's really a trivial part of the entire effort devoted to the process. So what you see is once you get over this activation energy of doing high-throughput phenotyping at all, you tend to collect a lot of time points because you can, and it might be useful. What I'm going to show you here, using an example from sorghum, which has been semantically segmented into leaves, stems, and panicles, is one benefit, one way we can use this time series data to help us. What I'm going to tell you about is a story that focuses on the sorghum. If you look at sorghum that is grown in temperate environments, it tends to be quite short, and that is controlled by 3 large effect genes, dwarf 1, dwarf 2, and dwarf 3. All of these genes have been identified and cloned. If you include plants that are quite tall, like this example you can see 2 of my students holding, which is probably 3 or 4 meters tall, it's easy to map these genes, but if you exclude extremely tall plants, it becomes impossible to identify these genes using just end-of-season measurements of plant height. Here what we had was time series measurements of plant height from the same plants, and this was enabled by the semantic segmentation. If we were just measuring the point from the bottom of the plant to the tallest point in the plant, the data gets a lot noisier. For any one of these points, we can identify if there are genes, natural variants in the population controlling variation in plant height using a genome-wide association study, which is conventionally shown this way with a Manhattan plot, where each of these points represents one marker. Its position on the x-axis indicates where it is on each of the 10 chromosomes of sorghum, and its position on the y-axis indicates how strong is the statistical significance of the association between variation in the genotype of this marker and variation in the target trait, in this case height. Because we have to do a lot of multiple testing correction, this dotted line indicates the threshold of statistical significance, and what you see is that we don't have any points above that line in this particular example. Now we can run this same kind of analysis at every single time point for which height was measured. We experimented with a number of different ways of visualizing this, from just showing lots and lots of Manhattan plots to one animated Manhattan plot that went from one to the other to the other. While these looked cool, it was often hard to identify patterns. So the solution we settled upon was what you see here. Each of these columns actually represents a single genome-wide association study. And then the color indicates whether there were any markers within this particular bin on the chromosome, which are above this threshold of statistical significance. What you can see is that while there were no strong signals identified on the last day of the study, we do identify signals at various points, different parts of the genome. And because height on one day is more correlated with the height on the days before or after that, when you identify a signal, it tends to sort of fade into statistical significance, get more significant, and then eventually fade out. With this analysis, we identified 2 of our 3 known true positive genes controlling plant height, dwarf 1 and dwarf 2. As well as many signals that were not dwarf 1 and dwarf 2. So I would argue this was a partial success. One real challenge is that looking at this, if I didn't know where dwarf 1 and dwarf 2 were, I'm not sure that I would pick the dwarf 1 signal as one of the first ones I would investigate. I might be lured away by, for example, this signal in chromosome 5 or the signal in chromosome 3. This is where it pays to collaborate and have conversations with talented statisticians. What I'm showing you here is the result of a collaboration with a very talented statistician named Yuhan Xu, who used to be employed at the University of Nebraska, has since been hired away to move to a university in Michigan. What he showed us and developed, really, or he developed the code that allowed us to do this analysis, was an approach that is known in the literature as functional principal component analysis, where you take a single average value for a large set of curves like this, which is this black line, and then you look at patterns of how different individual curves vary with respect to that original center curve, and then decompose these curves down into what these different patterns are. So what you see here is functional principal component 1, functional principal component 2, which are different ways that the curves can vary with respect to the average. And you can see that the vast majority of the variation we see in the raw curves can in fact be explained by just these 2 patterns of difference. So taller, shorter, or taller early and shorter later, taller later and shorter earlier. We can get values for each of our individual genotypes for each of these functional principal components. What you can see here is just what do the real curves look like for values on the 2 extremes of the spectrum for each of these. And then we can actually use those principal component scores as our phenotype for doing GWAS. When we do that, we are able to identify all 3 of our known true positive genes, dwarf 1, dwarf 2, and dwarf 3. We also get 3 unknown signals. This one on chromosome 3, we were actually able to validate by looking at another population, which mapped a QTL for height to this location. These 2 we were not able to validate, but the fact that 4 out of the 6 signals we were able to identify do have basis in the literature. We know that these are in fact regions of the genome controlling height, gives us a lot more confidence in the remaining 2 signals. So I mentioned that there are 2 ways that high-throughput phenotyping data really varies relative to conventional phenotyping. The second way that it varies is that it is much more likely to be multidimensional. And again, this comes back to essentially an economics or conservation of effort and resources argument. Once you've done all the work of collecting the sensor data from a bunch of plants, that sensor data can probably be used to measure many, many different properties of the plant. That's what you see here is an example where a single sorghum plant which has been semantically segmented, we're demonstrating different phenotypes which could be extracted from that single hyperspectral image. So once you have collected the sensor data and you've developed the software to extract the phenotypes, you're probably going to extract the phenotypes even if they are not necessarily the primary focus of your investigation, because it just costs you so little additional to add them, and they might be helpful. This situation is not unique to high-throughput phenotyping. We see similar things in human genetics where medical record data mining is enabling people to have many, many different phenotypes describing the same population of people. And similarly, if you're working with metabolomics or transcriptomic data and generate data from large association or mapping populations, you immediately end up with high dimensional data because if you do RNA-seq on a bunch of maze lines, now you have tens of thousands of phenotypes, each phenotype describing the expression of a particular gene in the genome. So this is work from another student in my lab, Jikai Liang. And because this is a relatively short talk, I'm not going to go into a lot of detail about the method. If you're interested, you can read about it in his molecular plant paper, which came out earlier this spring. But essentially what he was seeking is a way to actually leverage all this data from multiple phenotypes and be able to share data between correlated phenotypes What you see here is an example where you have many signals from different related phenotypes. All of these are linked to flowering time, but none of the individual signals on its own rises above the threshold of statistical significance. I should also mention this was again a collaboration with a very talented statistician, in this case, Yumo Chu, also formerly of the University of Nebraska, also hired away to a university in Iowa. So the output of the genome-wide phenome-wide association study is a list of genes linked to phenotypic variation in the population. And this is where we do our false discovery, control, and testing. For each of those genes, we also have a list of the particular phenotypes that GPWAS thinks the gene is linked with, although this is not rigorously controlled for false discovery. We face the same challenge we did with sorghum data, which is if we get some new results, how do we know if they're right or not? There's a more complicated answer to that using comparison across different populations, analyzed with different methods and how much concordance you get, which is probably the more statistically compelling. I'm going to give you the case study example, which is we can look at genes that have already been cloned and where there's already a known loss of function mutant. This is an example. The anthraeor1 gene is a classical maize gene involved in gibberellic acid biosynthesis. From studying the knockout, we know a lot about what this gene does. And GPWAS identified this gene as one likely controlling phenotypic variation in our study population and identified a set of phenotypes that were associated with it. If we do a comparison between these two, we see that there's actually a lot of agreement. So many of the phenotypes GPWAS identified line up with phenotypes supported in the literature from looking at the loss of function mutant or from other quantitative genetic studies with other higher powered populations. There also are things that line up with what we know about just the function of the gene. But I do want to highlight there are some additional phenotypes that GPWAS identified as linked to anthraeor1, where we could find no support in the literature. So these could represent false positives. Remember, again, we're not controlling false discovery at the level of individual gene phenotype pairs, but just which genes are controlling phenotypic variations all in our population. Or these could represent traits that are being controlled by anthraeor1, but which nobody thought to look for in comparisons of the mutant and wild type plant. I have a second example here, which is ligulus 2. And again, you can see good concordance between the phenotypes that were identified using GPWAS and the phenotypes supported by the literature. Now, when we look at the genes that I'm using as my control set here, so these are genes that with known loss of function phenotypes, there actually are a surprisingly small number of them, somewhere between 100 and 200, where somebody walked through a field, they saw a phenotype, they were willing to take the time and effort to map what gene is controlling this qualitative phenotype. But when you look at these genes, they really are different from the average gene model in the genome. They're more likely to make protein. They're more likely to be synthetically conserved. They tend to be longer and have more exons. And they are much less likely to exhibit presence-absence variation, which means that they tend to be in the genome of every single variety of corn, whereas many of the annotated gene models in the maize genome are present in some varieties of corn and missing from others. One of the questions I get when I show this result is maybe this is some sort of mark of essentiality. I can't test that question in maize because we just don't have enough cloned genes with known loss of function phenotypes, but using Arabidopsis data and a very similar analysis, we see that this is not the case. So if we look at genes with lethal phenotypes, with non-lethal phenotypes, or conditional phenotypes, essentially we see this same pattern of distribution where the overall set of gene models looks very different from the genes linked to any type of phenotypic variation. Similarly, if we go back to the genes identified with quantitative genetic methods like GPWAS or even conventional GWAS, we see the same pattern that the GPWAS genes are more likely to be expressed like classical mutants. They are more likely to be synthetically conserved. They seem to be under stronger purifying selection, and they're much less likely to be involved in presence-absence variation than the average gene. Same with GWAS genes. In fact, they show essentially overlapping patterns if we look at this distribution. The reason I think this is very exciting is that we are now working on methods to actually predict which gene models in the genome are more or less likely to be linked with phenotype. If we succeed, this will make it much more rewarding to do reverse genetics again, because it will be possible to identify which genes are likely to produce phenotypes. When I was a graduate student, I saw many people who really struggled because they were ordered up an Arabidopsis mutant, and they spent basically their whole PhD looking for some type of phenotype for that mutant, and they did not succeed. All right, with that, I'm almost exactly at 15 minutes, so I will thank you very quickly, and we'll be happy to take questions in whatever form that's going to take at this conference. Thank you very much. Stay safe. Good luck.